A series of new studies by researchers at NYU College of Dentistry highlights how e-cigarettes alter oral health and may be contributing to gum disease. The latest, published in mBio, finds that e-cigarette users have a unique oral microbiome, the community of bacteria and other microorganisms, that is less healthy than non-smokers but potentially healthier than cigarette smokers, and measures worsening gum disease over time. To our knowledge, this is the first longitudinal study of oral health and e-cigarette use. We are now beginning to understand how e-cigarettes and the chemicals they contain are changing the oral microbiome and disrupting the balance of bacteria, said Deepak Saxena, who led the research with Xin Li. Both are professors of molecular pathobiology at NYU College of Dentistry. Gum disease affects nearly half of U.S. adults over 30 years of age. Smoking cigarettes is a known risk factor for developing gum disease, but less is known about the impact of e-cigarettes, which vaporize nicotine and other chemicals, on oral health, especially the long-term consequences of vaping. The researchers studied the oral health of 84 adults from 3 groups: cigarette smokers, e-cigarette users, and people who have never smoked. Gum disease was assessed through 2 dental exams 6 months apart, during which plaque samples were taken to analyze the bacteria present. Changes to gum health: all participants had some gum disease at the start of the study, with cigarette smokers having the most severe disease, followed by e-cigarette users. After 6 months, the researchers observed that gum disease had worsened in some participants in each group, including several e-cigarette users. A key indicator of gum disease is clinical attachment loss, measured by gum ligament and tissue separating from a tooth surface, leading the gum to recede and form pockets. These pockets are breeding grounds for bacteria and can lead to more severe gum disease. In a study of the same participants published in Frontiers in Oral Health, the research team found that clinical attachment loss was significantly worse only in the e-cigarette smokers, not non-smokers and cigarette smokers, after 6 months. While all groups shared roughly a fifth of the types of bacteria, the bacterial makeup for e-cigarette users had strikingly more in common with cigarette smokers than non-smokers. Several types of bacteria, including Selenomonas, Leptotrichia, and Saccharibacteria, were abundant in both smokers and vapors compared to non-smokers. Several other bacteria, including Fusobacterium and Bacteroidals, which are known to be associated with gum disease, were particularly dominant in the mouths of e-cigarette users. An altered immune response: the researchers found that the distinct microbiome in e-cigarette users was correlated with clinical measures of gum disease and changes to the host immune environment. In particular, vaping was associated with different levels of cytokines, proteins that help regulate the immune system. In contrast, cytokines IL-4 and IL-1 beta were lower among e-cigarette users. IL-4 tends to be reduced in people with gum disease and increases after treatment, which suggests that certain bacteria in the mouths of e-cigarette users may be actively suppressing immune responses. The researchers concluded that the distinct oral microbiome of e-cigarette users elicits altered immune responses, which along with clinical markers for gum disease illustrate how vaping presents its own challenge to oral health. E-cigarette use is a relatively new human habit, said Scott Thomas, an assistant research scientist in Saxena's lab and the study's co-first author. Unlike smoking, which has been studied extensively for decades, we know little about the health consequences of e-cigarette use and are just starting to understand how the unique microbiome promoted by vaping impacts oral health and disease. This research was supported by the National Institute of Note. Content may be edited for style and length. Electronic cigarette use promotes a unique periodontal microbiome. Electronic cigarette aerosol modulates the oral microbiome and increases risk of infection. Electronic cigarette use enriches periodontal pathogens. Molecular Oral Microbiology. 2022. doi. 10.1111, OMI. 12361. Cite this page. New York University. https://ift. tt. 9z3 vjbg. Accessed February 22, 2022. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.